What has been the most interesting discovery in research so far?
Well, I think the most interesting discovery is something that we did as part of uh, being interested in developing a safer way of doing a hematopoietic stem cell transplant for crews. So we thought it was important to look at whether these things called telomeres. So telomeres are like little caps that are put on the end of all the chromosomes to protect them from getting kind of scuffed up from wear and tear. So they're a special kind of structured DNA that is very durable and protective. And it turns out that some research that had been done on SMARCL1 in tissue culture cells had suggested that not only was this protein important for just general DNA replication, it's also important for the telomere DNA replication. And so nobody had actually looked until we started doing this at whether this was true also for patients with SIOD. So this work was done mainly by Elizabeth Lipner in my lab and in collaboration with a researcher that actually does telomere studies in Canada and actually has a company that does clinical testing for telomere length. So we've collaborated with her and been able to get a number of samples sent to her and study a lot of SIOD patients, and we now know that all of the SIOD patients we've studied, I think we have now 7, every one of them has very short telomere lengths. And this is very significant because we know what the consequences of having short telomeres are in other diseases other than SIOD. So for example, if you have short telomeres, there's a tendency for your blood cell production to be adversely affected. And we think that this accounts for why crews, and to some degree Paisley as well, have reduced ability to make just their general blood cells by their bone marrow. This is probably accounted for, at least in part, by their having these shortened telomeres. And there's a lot of implications for this for clinically. So first, we now know that we probably are expecting that from here on, everybody that's going to potentially get a hematopoietic stem cell transplant for SIOD should have a bone marrow evaluation, and that we should anticipate that they will have reduced bone marrow production. And this is important. This will be an additional justification for doing a hematopoietic stem cell transplant. This will also help explain why certain drugs, when they're given to patients with SIOD, cause more bone marrow side effects. We've seen this with other patients where drugs that occasionally cause you to, for example, have a low count of a particular blood cell, in the SIOD patients it's much more common. We now think that we know why this is because of the short telomeres. And there actually are some drugs that we may be able to see if we can use them to increase telomere length, and perhaps in the patients that can't get hematopoietic stem cell transplant, we might still be able to improve their bone marrow function. So I think that's all pretty exciting. And then the most important thing, though, for Cruz is that once we knew that he had short telomeres in his blood cells, we could devise a much safer protocol for him to get his hematopoietic stem cell transplant, because there are a number of diseases that also have short telomeres for which they also have reduced bone marrow function, and they have developed safe ways of preparing the bone marrow to receive the new stem cells. So we actually, with Dr. Alice Britannia, developed a a way of doing this in a safe manner, and that's actually the protocol that he received. He got a low dose of radiation and some special drugs at doses that we thought would be safe for him. So I think we got a lot out of just that one study, and we both got some understanding about the disease and its mechanism, but also some really clinically important information. But I want to say, though, that we just think the short telomeres is not the whole story. All these other patients with these other genetic conditions that lead to short telomeres, they do not have the kidney problems, they don't have the T-cell immunodeficiency, they don't have short stature. So there's obviously something still about SMARCL1 protein that we need to figure out.